Hello students, this is Molecular Basis of Inheritance Part 5. Today we are going to discuss about DNA replication. So what is DNA replication? The process in which the DNA molecule produces exact copy or a replica of itself, exact Xerox of itself, then that process is called as DNA replication. In eukaryotes, the DNA replication takes place inside the nucleus, and DNA molecule, it regulates, it controls all the activities of the cell. So all the activities, they are controlled and regulated by the DNA molecule. DNA molecule has a unique structure. It is double-stranded, and it is a helical structure, helix-like structure, right? And because of that, it is able to control the synthesis of other molecules. All the molecules which are present inside the cell, their synthesis is controlled by the DNA. It contains the genetic information. So when the cell gets reproduced, at the same time, DNA molecule gets duplicated equally to the daughter cell. In molecular biology, DNA replication is the biological process of producing two identical replicas of the DNA from one original DNA strand or one DNA molecule which is original or parental DNA. So that DNA replication occurs in all the living organisms, acting as the most essential, most important part of biological inheritance. Now we know that DNA is a carrier of genetic information. It carries all the genetic information of that particular living organism. Okay, so DNA has to perform two important functions. First function is heterocatalytic function. Another is autocatalytic function. First is hetero. Hetero means different. So what is different? Auto means same, or auto means similar. Okay, so or self. So what is heterocatalytic function now? When DNA directs the synthesis of chemical molecules other than itself, other than the DNA itself, then such functions of the DNA are called as heterocatalytic functions. Now, for example, there are two examples which are given: synthesis of RNA and synthesis of protein. Synthesis of RNA is a process that is called as transcription, and synthesis of protein is called as translation. So we are going to study this transcription translation part in the next lectures or in next sessions. So we are not going to talk about this right now, but you have to remember, in the transcription and translation process, what DNA is synthesizing? DNA is synthesizing RNA molecule. Okay, so the process of synthesis of RNA or formation of RNA molecule is called as transcription. Okay, and another is protein, not the DNA, but from the DNA molecule we are getting the protein. Okay, so that type of process is called as translation. So it is a heterolytic function. Okay, now what is autocatalytic function? When the DNA directs the synthesis of DNA itself, then such function of DNA is called as autocatalytic function, that is replication. Today we are going to discuss the replication process. Now, what is replication? In the replication, we talk about when a DNA molecule or the DNA strand forms its exact replica or its exact Xerox copy or its exact copy. Okay, so when that occurs, when a DNA forms or synthesizes a DNA molecule, okay, the same molecule when get formed, then that type of process or that type of function is autolytic function, and that process is called as replication. So DNA get formed or synthesized by a process that is called as DNA replication. So let's have a look. Let's study what is DNA replication in brief. So the process by which the DNA duplicates itself is called replication process. Through replication, it forms two copies that are identical of it. Okay. And in eukaryotic organisms, the replication of DNA takes place only once in the cell cycle. It occurs in the S phase. S stands for synthesis phase of interphase in the cell cycle. So DNA replicates through a mode of replication, and that process is called as semi-conservative replication process. The model of semi-conservative replication was proposed by Watson and Crick, these two scientists, on the basis of the anti-parallel and complementary nature of the DNA strands. The process of semi-conservative replication is shown. Now see, this is one strand. Now these are the strands which are the old strands, okay? And the newly synthesized strands, one strand is discontinuous and another strand is continuous. So these are the newly synthesized strands, or they are daughter strands. So semi-conservative replication is that type of replication when out of these two strands of DNA, one strand is old and another strand is new one. One strand is parental DNA strand and another is daughter or newly formed, newly synthesized DNA strand. Again, same with the other strand. One is old strand, that is parental strand, and another is the newly synthesized strand. Okay, so that type of process is called as semi-conservative replication. Out of these two strands, one is old and another is new, and that type of model or that type of semi-conservative replication process was proposed by Watson and Crick. So let's have a look what are the steps and what is semi-conservative replication of DNA. So when we discuss about DNA replication process involves lots of steps. Okay, first is activation of nucleotides, that is called as phosphorylation. Second is origin. The nick or the incision takes place, and from that point the DNA replication gets started. And another point is the synthesis of strands. Now there are two types of strands. They are formed on the DNA strand as this DNA helix is, it is supercoiled and it is anti-parallel nature, so it gets relaxed. That coiled DNA is uncoiled at a certain point, and then it is replicated. A leading strand get formed, which is in continuous manner. The newly synthesized DNA or that daughter DNA, it gets formed in a continuous manner. That type of strand is called as leading strand. Another strand get formed simultaneously, which is not continuous but it is incontinuous, and it gets formed in the fragments. Those fragments, they are called as Okazaki fragments. Then the DNA ligase plays its role. DNA polymerase, gyrases, SSBP, that is single strand binding proteins, number of enzymes, they are involved in the DNA replication process. And as we are going to discuss the replication process in the stepwise, like in step 1 the activation of nucleotides takes place. In step 2 there is formation or origin, or there is formation of Y fork. In the third step the strands get formed. So for our convenience, we are studying the DNA replication process in stepwise manner. In actual uh, in actual situation which occurs, the DNA replication which occurs inside the cell, it occurs in a continuous manner. Okay, when there is activation of nucleotides, origin or the point of origin or the unwinding of DNA is got started. Okay, so in that, without any chaos, number of processes, they occur at the same time in the cell. So let's have a look. Without animation, it's highly impossible to try to understand the DNA replication process. Now in the next part I'm going to tell you or describe the, at a glance we are going to study that what exactly happens inside the DNA. We are just going to start till the leading strand. We are not going to discuss the lagging strand, as the lagging strand, that part is a little difficult, or that part we will discuss later. So let's have a look. Let's study DNA replication in the real-time animation. This is DNA replication. See, this is DNA molecule, which is a twisted molecule. Can you see that? Yes. Now this twisted molecule is getting untwisted, and this is a ladder-like structure. These are the ATGC base pairs. Two strands of DNA, and in between these two strands, these are the base pairs which are present, right? Now we know that DNA double helix is anti-parallel because it has one strand 5' to 3', another strand 5' 3' to 5'. Okay, that's why it is anti-parallel in nature. Now see, this double helical and anti-parallel structure is getting separated. This is helicase enzyme. It forms a replication fork. Helicase moves forward, it unwinds, it separates the two DNA strands. Out of these two DNA strands, one strand, concentrate on one strand. RNA, another enzyme, that is primase enzyme, is attached at one point. It forms RNA primer. RNA primer is required for the attachment of another enzyme, that is DNA polymerase. Now see how beautifully the DNA polymerase gets attached to the RNA primer, and these are the active nucleotides. Phosphorylation, by a process phosphorylation, these active nucleotides, they get attached, they are getting added, right? And the DNA polymerase is moving forward, the bases they are added, and the leading strand get formed. Can you see that? The replication fork, the DNA polymerase, how beautifully it moves forward. It forms a continuous strand. That strand is called as leading strand. Okay, so that's all about this animation. The another part is about lagging strand, that we are going to discuss, as I have already mentioned. That part we are going to discuss in the next section, or in the next part. So in today's session, we have studied how the DNA replication takes place inside the cell with the help of beautiful animation. So that is not enough. Actually, you have to read the textbook. So read the textbook, and we have not completed the DNA replication yet. I'm going to discuss each and every step which is involved in the DNA replication process in detail. We are going to talk about, but not today. So that's all for today's session. Thank you.